Crossing over, random assortment, and fertilization all lead to genetic diversity and variation. So when we think about meiosis 1, sometimes we say we take the chromosomes, we mix them up, and we separate the homologs, or the homologous pair. So what does it mean to mix them up during meiosis 1? Well, during prophase, we have this synapsis occurring. And during this synapsis, we can have what's called crossing over, which results in recombination. So we have sister chromatids, and sometimes the sisters can kind of weave as the homologs are combined here. The sister chromatids can kind of weave with each other, and there can be double strand breaks. So you still, instead of just having maternal or just having paternal, you can actually create new combinations of chromosomes that contain both maternal and paternal within the same chromosome. We also can have independent or random assortment. During metaphase 1 of meiosis 1, all of these now mixed up homologous chromosomes are going to line up at the metaphase plate, but the way they line up is random. You don't always just have paternal on one side and maternal on the other. You can have a combination of paternal and maternal chromosomes on each side. So if we look at that, if you would, for example, have 3 chromosomes, you could have all paternal, all maternal. You could have 2 paternal and 1 maternal, and then the other side would be the opposite. So with 3 chromosomes, you can end up with these different combinations of which are on which side because they independently assort or randomly align during metaphase. So when we start thinking about the diversity here, different organisms have different numbers of chromosomes. What we're looking at here is 3 pairs of chromosomes. So to find how many different combinations we'd have 2 to the 3rd power, which gives us 8 possible combinations for 3 chromosomes. But as you add chromosomes, we start getting more and more possible combinations. So if we think about humans having 23 pairs of chromosomes, what that results in is over 8 million possible combinations, which is pretty incredible. So when we start thinking about that, there are over 8 million different combinations of sperm that can be made from the same 23 pairs of chromosomes. Now, when we start thinking about this, as we keep going further, it gets more and more complicated and we have more and more diversity. Because during meiosis, we don't just have the sperm, we also have the egg. So through independent assortment, we can have over 8 million possible combination of sperm gametes. We could have over 8 million possible combinations of egg gametes. But then when we start randomly fertilizing these and take any random egg with any random sperm, we end up with an exponential growth. So ultimately, between 2 individuals, you could have over 70 trillion possible combinations to make zygotes. So another way to think about this is, again, if we're talking about one trait on one chromosome, that would be 4 possible. So when we think about Punnett squares, the outside are the possible gametes, the inside are the possible zygotes. So there's 4 possible zygotes that can be created from a single chromosome. By simply adding one more trait or one more chromosome, we can get 4 possible gametes and 4 possible gametes, resulting in 16 possible zygotes or offspring. And again, every time we add another chromosome, we exponentially increase the number of possible zygotes. So then when we start talking about 23 chromosomes, that's how we get over 70 trillion possible zygote combinations. So again, as we look at this, 3 major ways we get genetic diversity: through recombination and crossing over, which happens during synapsis of prophase during meiosis 1, random assortment, which occurs during the alignment at the metaphase plate during meiosis 1, and random fertilization, which occurs during sexual reproduction. So, crossing over, random assortment, fertilization all increase variation.